It's great to have you, Thomas. Thank you for joining me today. If you could introduce yourself a little bit for our audience, that would be great.
Okay, my name is Tomas Rube. I'm currently at University of California in Merced. I got my PhD in theoretical physics, and then I transitioned into computational genomics. So my work tends to be in the interface of genomics and machine learning and biophysics. For the work we're going to talk about today, this actually started when I was a postdoc in Harman Bussemarker's lab at Columbia University. And now I'm just about to start my own lab here at University of California in Merced. So that's me.
Nice, thank you. So the reason I invited you was to talk about your recently published paper, which was predicting protein-ligand interactions using machine learning. So I guess we should start by maybe having you introduce that paper a little bit. What was the idea behind the paper? What was the overall themes?
Yeah, so our main focus is to understand the transcription factors and how they work. So first, what are transcription factors? So transcription factors are proteins, and they bind specific targets in the genome. And when they do that, they regulate the transcription of nearby genes. And they're really powerful. They're really, really important for cellular information processing and decision making, because a single transcription factor can bind many targets in the genome and therefore regulate many genes. So by tuning the activity of a transcription factor, the cell can regulate the whole battery of downstream genes. And so understanding how transcription factors work and how they find a target and what genes they regulate, that's a really essential question in molecular biology. So how do transcription factors find their targets? Well, it's complicated, the many contributors. One important factor is the sequence that it binds. And so transcription factors have DNA binding domains, and these have evolved to bind specific sequences. And so if that sequence is somewhere in the genome, there's an increased likelihood that the transcription factor will bind there. And so typically there's one sequence that's bound really, really strongly. But you can also have mismatches compared to this sequence, and you can still bind, but more weakly. And it turns out that these weak, low affinity binding sites, they're actually really important. They're really common, and they can be functional. They can be 100-fold below optimal strength and still be functional. And if you mutate them to be stronger, you can actually mess up the regulatory programs.
Right.
And so the big problem we tried to solve was, given a sequence, can we predict the strength of the binding site, the binding? And not just getting the top sequences, but but all across the affinity range. That was the kind of the big problem we're trying to solve. And in practice, what does that mean? Well, it means that you give me a sequence, and I have my equation, and I plug the sequence into the equation, and I give you back the strength. Yeah? So we're doing machine learning to learn some kind of mathematical model that predicts strength given sequence. So that's in practical term, what it means. Now, transcription factor binding is it's really complicated, actually. Now, the sequence of the binding site is really important, but other factors also contribute. So, for example, transcription factors can form complexes with other transcription factors, and then they can bind together to the DNA. In doing so, they can read out a longer sequence and can be more specific in what they bind. The chromatin is also really complicated. The DNA can be chemically modified, it can be packed away in nucleosomes and so forth. And so it's really quite complicated problem. So there's a big push in the field to not only understand how transcription factors bind specific sequences, but to understand these other variables as well. And so that's the big question we're trying to understand here. And so if you zoom in a little bit more, what is it we're actually trying to do here? Well, okay, so we're trying to learn these mathematical models that can predict the binding strength as a function of sequence and other variables. And there are two main approaches to getting at this question. One is to take the in vivo approach. That's to say, you map where in the genome the transcription factor binds. And this is really powerful. It's really good for figuring out what the targets of the transcription factor are. But the genome is also very complicated. And so given that you know that it binds somewhere, it can be really hard to tease out what the causal mechanisms that actually made it bind there, or what it is. An alternative approach is to profile transcription factor binding in vitro. Okay, what does this mean? What this means in a test tube. So what you do is you take your transcription factor, you take some DNA sequence, and you mix it together, and then you measure the strength of the interaction. And so this is kind of a complementary approach that's really powerful for characterizing the intrinsic properties of the transcription factor. Cool. And so this is the approach we're taking in this study. And if you zoom in a little bit more, so over the last decade or so, high throughput sequencing has really revolutionized how measurements are being made. And in particular, a big class of assays called Celex sequencing assays have proven really powerful for characterizing how protein find their ligands. And how do these assays work? Well, the starting point is a pool of randomized DNA. Okay, so you have a little test tube, and you can synthesize random DNA molecules. So because there's so many molecules, even a tiny tube, it can be billions and billions and billions of unique molecules. It can easily contain all 15 base pair sequences. So it can easily contain all good binding sites and all bad binding sites. And so the idea of this approach is to use this very complicated, complex library of random molecules and profile how they all bind to the transcription factor. And it's really powerful because it's unbiased. You don't need to know what the transcription factor binds a priori. You can just profile them all. So that's the starting point of the approach we're taking. And so then, so how do you actually use randomized DNA to characterize transcription factor sequence recognition? Well, so the next step is you mix in the transcription factor in the tube of random DNA. And it will bind to some sequences, not to other. And so then you separate out the DNA that was bound. And so things that are really bad binders won't be there. Things that are so-so binders will be there, and the great binders will definitely be there. And then you can repeat this cycle over and over again. So you amplify whatever was bound, and then you do another round of binding selection, and so forth. This is a classic experiment called Celex. So
how do you get rid of the unbound DNA?
Yeah, that's a great question. There's a number of ways you can do that. One way is to separate them on a gel. Right. So the bound DNA and the free DNA, they migrate at different speeds on a gel. Another way is to use magnetic beads. So you put the transcription factor in a magnetic bead, and then you can just wash off whatever is not bound.
And you can just attach the transcription factor to the bead? Exactly,
yeah. Okay. Exactly. So the transcription factor is hooked up to the bead, and then it binds grabs some to some DNA, and whatever it doesn't grab on is unbound. Nice. And a third way is to use microfluidics to mechanically trap whatever is bound. So those are some ways. And so the sequencing approach, what you do is you extract a little bit of DNA from the initial pool and then from all the selected pools. And so modern high-throughput sequencing can read millions and millions and millions of molecules. So you can get a huge amount of data. It's really powerful. And so the idea is that the sequencer gives you a text file. If you look through a text file, initially, if you look at the initial pool of DNA, it would just be a random text, random strings of letters. But as you go through the selection round, you will start to see some sequences becoming more frequent. And this is because the transcription factor is binding those sequences. And so the idea is that by analyzing how this pool of random DNA evolves as you select for binders, you can infer what sequences the transcription factor binds.
Right. Yeah.
Now, so you get these text files with 100,000, a million sequences in them. And they're only useful insofar as you actually have an algorithm to take the text and then turn it into knowledge about a transcription factor, a predictive model that can predict how any sequence would have been bound. So that's the problem we're focused on here. And solving this problem qualitatively is pretty easy, but if you want to be really quantitative and accurate, it's actually a pretty hard problem. So that's the problem we solved in this paper.
Okay, what are the issues that you said at the end? How is it easy to do it qualitatively but not quantitatively?
Well, it's a number of issues. So one big issue is that the data tends to be extremely noisy. And so why is that? Well, so I like to think, if you think of a DNA library as a huge bag with lots of slips of paper in it, and what the sequencer does is that it randomly pulls out some of this piece of paper and reads them. Right? And so say that you have a huge bag and there's 1 billion pieces of paper in it, and the sequencer reads 1 million of them. Right? So 1 million is a pretty big number, but each little paper only has a 1 in 1,000 chance of being read by the sequencer, right? And so the chance that you'll read any sequence twice is basically 0. And so given that you have this big bag and you pull 1 million, I pull 1 million, you're mostly going to get different sequences. And so which sequences we see is basically determined by random noise. Now, as you select more and more, you kind of zoom in on a subset of the sequences, but typically the number of sequences is still so big, so it's just random chance which sequence you see, and so that's a big challenge. Now, this is a pretty common problem, but it's well known in statistics that even though you have very noisy data, if you know where the noise is coming from and you model it, and you have a lot of noisy data, you can still overcome the noise and get really accurate models. So that's kind of what we did. So that's one challenge. Another challenge is that, so remember, the goal here is to learn this equation, this binding model that predicts binding given that you give them a sequence. On the other hand, we have the sequencing reads, which is the data we're trying to learn from. And the relationship between these two is actually quite complex and non-linear. So some of the things that make it kind of complicated is that you have multiple rounds of selection, and the binding signal kind of accumulates across rounds.
Right.
You can also have artifacts such as something called binding saturation, which makes the relationship between strength and binding nonlinear. Given that you have a single piece of molecule, binding probe, a piece of DNA, the transcription factor can bind at different positions in it and so forth.
Right.
And so if you want to be really quantitative, you need to account for all these things. If you just want to get the simple answer, you can just look for substrings that are enriched, and it kind of works decently well, but if you want to be really quantitative, you need to account for all this.
And how do you go about accounting for those things?
Well, so we were inspired by a machine learning method called deep learning or neural networks. And so this is a really powerful machine learning method. It's a family of many methods, and it's been extremely useful for a wide range of problems. And the idea in deep learning is that there's something you want to predict. Say you're Amazon, you want to predict how well a product, if a user is going to buy a product, and you have some predictors, maybe the past purchases. And so the idea in the deep learning approach is that you start with the predictors at the top, and then you feed them into a bunch of artificial neurons. This is inspired by the brain. And then this first layer of neurons feed into next layer of neurons, and next layer of neurons, and next layer of neurons, and finally the last layer of neurons produce an output, which is the prediction. And then in deep learning, you optimize the connections between the neurons to get a really good input or predictor to output, which is the variable you're interested in, the good relationship. So that's the idea of deep learning. So we're kind of inspired by this, in that we created a multi-layer model that tries to predict binding given sequence. Now, a drawback of this kind of deep learning models is that they're really hard to interpret. So given that you look at a specific neuron, it's basically impossible to know what it means. And so what we did is we wanted the different layers correspond to a specific mechanism in the causal chains going from sequence to binding. So for example, the first layer to be the binding model that we're actually interested in that predicts the binding strength. The next layer wanted to correspond to the assay, repeated selection, and the final layer corresponds to the sequencing. And it turns out that by doing some mathematical modeling and imposing domain knowledge on the lower layer levels, where you model the assay and the sequencing, then you actually, if you do it in a rigorous way, you can actually force the top layer, which is the binding model, to be really accurate. And so that's kind of waving my hands, telling what we did. So you can kind of think of it as a kind of a flavor of deep learning, perhaps, that's kind of really constrained by domain knowledge and modeling.
Okay,
so that's like this.
Took, I guess, took a lot of data from these Celex runs, and is that what you used to train your model?
Exactly. Yeah. So the input in the top is the sequence we got from the Celex experiment, and it's trying to predict which round it came from. Basically, is this from the input or later rounds? And so you can think of it as a classification problem that way. And then we optimize the whole model. And it turns out if you optimize the whole model to do that particular classification problem, and you also make the lower levels be accurate, then you force the top layer to correspond to the quantitative binding strength.
Right. Yeah.
And so that's kind of the second challenge we need to overcome, like how you actually model these experiments in a quantitative way. Another big challenge is that there are many types of Celex experiments. And so I mentioned earlier that if you're interested in where transcription factor binds, like a really important variable is the target site. But the many other things that are important, like cofactors or the chemical modification of the DNA and many other. And so there's this big push in the community to understand how these higher order effects contribute to transcription factor binding. And so variants of Celex have actually been developed and deployed already to profile many other variables that impact transcription factor binding. And so a big challenge that we wanted to overcome was to have one algorithm that worked on all of these different kinds of Celex data. And even beyond transcription factors, there are many other protein classes that also recognize sequence. For example, RNA binding proteins recognize RNA sequences. If you think of protein-protein interactions, for example, kinases, they phosphorylate peptide targets in a way that depends on the target sequence. So sequence recognition is a much bigger phenomenon, and assays that are similar to classical Celex sequencing have been developed and deployed to characterize this. And so we wanted to have a kind of a flexible algorithm that could solve all of these problems. So that's another challenge that was pretty difficult. And then there's like a fourth challenge, which is that using Celex so far, you've only been able to get relative measurements of binding strength. So if you give me two sequences, I can say, well, sequence 2 is 5 times stronger than sequence 1. But if you ask a biochemist, so how can you measure the strength of these binding sequences? They will not tell you that. But the biochemist would measure the dissociation constant.
Right.
That measures the absolute strength of the binding. So he would say, well, sequence number 2 is a 2 nanomolar strength, and sequence 1 is a 10 nanomolar strength. So it's weaker. So it would be nice if you can get like absolute binding strengths out of high throughput sequencing data. And initially, actually, I thought it would be totally impossible because from sequencing data, you can only kind of estimate the relative frequency of different sequences.
Yeah.
And so it's kind of explaining how it works is kind of subtle, but we figured out a way of modifying the experiment and use machine learning. Yep.
So using your model now, you would not need to use the Celex method, or would you sort of use it together?
Oh yeah, you also need to use always need to have the experimental data, right? But a single random selection seems to work really well. So we say just do a single random selection and sequence the input bound and unbound, and then you can get absolute KDs. And another good thing about that is that they also correct for binding saturation, which is kind of annoying artifact you need to deal with.
So how many runs would you normally do in a Celex without using your model? Like what is the improvement that you're making?
Can you say it again?
What exactly is the improvement that the model makes on the current method?
Well, there kind of a number of improvements. One is that our method can really leverage all Celex rounds, right? And so remember the Celex, you start with random DNA and then you select multiple times. And the early rounds, they have mostly actually low affinity sequences because they're way more low affinity sequences than high affinity sequences. So they're really common in the early rounds. And later rounds, they have mostly high affinity sequences. And so if you want to learn both about high affinity sequences and low affinity sequences, it's nice if you can use all the data. And so our method systematically integrates information from all the rounds to get models that are accurate over a wider affinity range. So that's kind of one improvement. Another improvement is that our method can use different alphabets. So if you think of standard DNA, we have ACGT, but turns out it's kind of handy to be able to use different alphabets. For example, if you're interested in how DNA methylation impacts binding, or what's DNA methylation? Well, the bases can actually be chemically modified, and so this is quite common and can impact binding. And so if you just have the ACGT alphabet, you can't really encode such modifications. And so by using extended alphabet, it can encode methylated DNA or other chemical modifications, and also extending the binding model to have this. You can get binding models which are, we call it methylation aware. As I said, They can not only predict binding to regular DNA, but chemically modified DNA. And having an extended alphabet is also useful if you're interested in interactions with RNA or peptides and so forth. So that's kind of another improvement. Another kind of feature of our approach is that you can jointly analyze multiple experiments. And this is also kind of useful for many applications. So for one application is, well, maybe the same transcription factor has been profiled by many labs, and you would want to have a consensus model that optimally explains all data. Maybe the different labs have different artifacts, and so if you learn from all of it, you'll get rid of the artifact and just learn what's real.
Right.
Another application is if you do this kind of profile DNA methylation. In that case, you typically have a normal experiment and a method experiment. And so being able to learn from multiple experiments is actually really critical for that. Or if you have different transcription factor concentrations or treatment times and so forth. And so being able to learn from multiple experiments is, I think, really important. That's a direction the field needs to move in. And then it's also these absolute KDs I told you about before. And it also comes from the ability to integrate multiple sequencing libraries in a really systematic way.
Yeah, that's incredible, the KDs. How certain is the accuracy of those KDs?
Yeah, so we profiled a transcription factor called distillus, and then compared the prediction from the model to low throughput measurements using EMSA, and they're really on the line now. So you don't seem to lose anything by using like a randomized library and machine learning compared to profiling individual.
Oh, wow.
Yeah,
Awesome.
but that of course depends. It requires them all to be accurate, right? And so if you have a much more complicated systems where the model is not accurate, then of course it could be less accurate. But for transcription factors, it seems to work really well.
Yeah, that's so cool. Is there much more that you think we've not touched on? I mean, I'm sure there's plenty more, but is there anything you wanted to add?
Yeah, no, I think we've covered most of it. I think really the ability to, our bottom models seem to be a little bit better, in terms of accuracy over a wider range. If you're interested in using them, you should go to motifcentral.org and check them out. But I think the big takeaway of the paper is that we should think more about how we can integrate complementary experiments, especially as we look at higher order features of how protein finds the ligands, and also that doing a little bit of mathematical legwork when you model the assay can really facilitate new things. And so, for example, the KD sequence, the reason we could come up with it was because we could really model the assay really accurately and understood how it works. And so I think that those are some of the bigger takeaways if you're a modeler.
Yeah, that was great. Thank you very much for joining me today.
Thank you. It was great talking to you, and thank you for the invitation.
Yeah, of course. It's been a pleasure. Thank you.